We will now have the keynote lecture by Professor Ashok Venkitaraman. Professor Venkitaraman is the Director of MRC Cancer Unit, University of Cambridge, UK. His laboratory studies chromosomal instability in cancer pathogenesis and treatment. He is associated with India Alliance as a member of the Intermediate and Senior Fellowship Selection Committee member. Ashok, please.
It's a real pleasure to be here this morning to be speaking to you. And what I'd like to do in the next 30 minutes or so is to share with you some insights from my lab over the last few years concerning the problems that we've been working on for more than 2 decades. But before I start talking about my science, I'd like to also take this opportunity to congratulate Shahid and his colleagues at the India Alliance for their 10-year anniversary. As I think all of us in this audience will agree, it's really been tremendous that the India Alliance has, within a relatively short space of time, been able to make such a wonderful impact on cultivating Indian science, particularly through funding promising young investigators. And it's been a real privilege for me to be associated with the India Alliance in a very small way over the last several years. So with that said, what I'd like to do is to talk about the work of my lab as exemplified in the title, From Cancer Susceptibility Mechanisms Towards Early Intervention in Cancer. So as many of you know, the work that we have been doing in Cambridge over the last 2 decades has really been focused around, if you will, a dream. And that dream is whether in cancer, as in many other chronic diseases, there are opportunities for early intervention. What do I mean by early intervention? For me, the scientific issues are encapsulated in these 3 questions. First, can we understand, before they develop cancer, which individuals amongst the population are most susceptible to the disease? Once the disease begins, can we understand which early lesions are most likely to progress? Because as we well know, not all of them will. And finally, with this information, can one begin to halt progression early, or even potentially in the future to prevent or delay the onset of cancer? And these questions obviously need a good understanding of the conspiracy, if you will, between environmental factors and genetic risk causing susceptibility to cancer, a good understanding of the key molecular and cellular events during early steps in oncogenesis, and finally, enabling technologies to be able to try to take some of this information towards clinical application. My own work started on this problem roughly 2 decades ago, trying to answer or address a simple clinical observation that I came across when I was training in medicine here at the Christian Medical College in Vellore. The observation is that in most common sites of carcinogenesis, very few incipient neoplastic lesions actually progress to invasive malignancy. And this, of course, implies that there must be multiple homeostatic mechanisms, both cell-autonomous as well as non-cell autonomous, whose purpose it is to eliminate or prevent the progression of these early lesions, and mostly they work very well. But sometimes they fail, and that is when incipient malignancies are able to proceed towards invasive disease. So our somewhat simplistic thinking when I started my laboratory off at the MRC, Laboratory of Molecular Biology in Cambridge, was to ask whether looking at families or people with inherited early onset cancer syndromes would give us clues as to what these mechanisms were that prevented cancer. The rationale being that in these individuals, the genetic mutations that conferred early onset cancer predisposition must in turn affect one or more of these homeostatic mechanisms. We were very lucky because at the time we started on this problem, there had been major advances in understanding the genetics of cancer susceptibility, particularly in breast cancer. What I say here applies to most epithelial cancers, although I'll illustrate it with breast as an example. Only about 5% of epithelial cancer cases exhibit a strong familial pattern of inheritance. The majority are sporadic. We now know that germline mutations contribute to high penetrance risk for many of these familial cases, but not all. Even in breast cancer, which is arguably one of the best studied epithelial neoplasia, we only understand about half of the familial risk. Only about half. Can you believe it? After 20 years of study. But what we do know is that certain high penetrance mutations affecting genes like the so-called breast cancer genes, BRCA1 and BRCA2, are important predictors of susceptibility to familial cases of breast and ovarian cancer. And what we now also know is that it's not just germline mutations affecting these genes, but also a number of somatic mutations either affecting these genes or affecting the pathways, the cellular pathways that they work in, that contribute to the majority of sporadic cases of cancer as well. Regarding BRCA1 and BRCA2, which I'm going to speak about now, we know that inheritance of heterozygous mutations, in other words, mutations affecting a single one of the two copies, suffices for predisposition not only to breast cancer, but also to cancers of the ovary, pancreas, prostate, and many other tissues. We know that these are high penetrance mutations in that carriers exhibit a very high cumulative risk of developing these types of cancer by the age of about 70 to 80 years. Interestingly, it's become apparent quite recently that mutations affecting these germline cancer susceptibility alleles may account for a significant fraction of many human cancers. So this is a recent study from Wash University where they looked at over 10,000 cancers of 33 different types. And what you see here is a plotting of the percentage of cancers at various sites that can be accounted for by pathogenic germline variations. And what you see is that mutations affecting the breast cancer genes, BRCA1, BRCA2, a partner of BRCA2 called PALB2, account for a very high frequency, relatively speaking, of cancers of the breast, the ovary, the pancreas, the esophagus. In other words, several relatively common epithelial sites. So what I'd like to share with you over the next 20 to 25 minutes is some of the work that we've done in my lab trying to use these pathogenic germline susceptibility alleles to exemplify opportunities for early intervention in cancer. In other words, through understanding how mutations in these genes, like BRCA2, confer cancer risk, and by understanding the processes involved in carcinogenesis, can we actually begin to devise strategies, at least in this high-risk population, for early detection, risk stratification, and potential early intervention. So to think about some of the recent data from my lab, let me begin by telling you what I suspect every single person in the audience will know, which is the so-called Knutson-Hidden paradigm for tumor suppression. So please bear with me. As I said, I'm sure this is familiar to virtually all of you. The idea is that for germline inheritance of susceptibility alleles for cancer, tumor suppressor alleles, that you inherit a single copy of a mutant allele. The second copy is lost in certain cells. This is called, confusingly, loss of heterozygosity, but it actually means mutations affecting both alleles. And this loss of heterozygosity event, the second hit, if you will, is what promotes carcinogenesis. And when we started our work, it was believed that this LOH event, the loss of that second copy, was vital for carcinogenesis to begin. We now know through the work of my lab and many others across the world why this may be the case. We know that when you inactivate BRCA2, both copies of BRCA2 by loss of heterozygosity, cells exhibit spontaneous chromosomal instability. And it's literally 20 years ago to this year when my lab published this work showing that when you knock out both copies of BRCA2 in the mouse germline, you get these grotesque chromosomal abnormalities, both of chromosome structure and number. And you see some of them highlighted by arrows here. Those of you who look at chromosomes will immediately realize that the vast majority of these lesions are of the so-called chromatid type. In other words, there are breaks affecting one of the two sister chromatids, or these grotesque structures called radial chromosomes, triradials or quadriradials. And these are pathognomonic of many cancer susceptibility syndromes, such as, for example, Bloom syndrome. And they led us, when we found this at the time, to suggest that genome instability, in a manner similar to cancer susceptibility syndromes like Bloom's or Fanconi anemia, was what underlay, if you will, carcinogenesis following BRCA2 inactivation. We now know through the work of many labs that BRCA2 inactivation disrupts a critical form of repair of DNA lesions. This is DNA repair by homologous recombination, which is a mechanism that mammalian cells use to deal with double-stranded DNA breaks. So when you get a break, which is typically flush-ended, you get nucleases that resect one of those strands, leaving a long overhanging single strand of DNA. BRCA2 then catalyzes reactions that bring to this single-stranded DNA substrate the recombination enzyme RAD51, which is the mammalian homologue of bacterial REC-A, which many of you will be familiar with. And RAD51 forms an ordered helical nucleoprotein filament on the single-stranded DNA, and that structure enables invasion of the homologous duplex, the templated strand, and that leads to the strand exchange events that lead to error-free repair by homologous recombination. You immediately see when I describe this process why you get chromatid-type errors in BRCA2 deficient cells, and you also realize that this process is vital in all dividing cells. It can only work during the S and G2 phases of the cell cycle when you have a perfect templated copy against which to perform the repair. So when BRCA2 is inactivated, this process is gone, and what you're left with are error-prone processes that repair double-strand DNA breaks by mutagenic events leading to mutations, and we think carcinogenesis. We now understand how BRCA2 promotes RAD51-mediated homologous DNA recombination in some detail. And I'm not going to take you through all of the work, but just highlight a few of the steps that we have identified in my lab. So this is BRCA2, and what you see here is that it's a ginormous protein. My postdocs still curse me because it's 3,500 amino acids long. No one can purify it as a single isolated protein. It's extremely difficult to do biochemistry or structural work with, and we, like many others, have therefore been looking at pieces of the protein rather than the full-length molecule. So one of the first structures that we solved with Luca Pellegrini in the Department of Biochemistry at Cambridge going on over a decade ago was the structure between these 8 repeated sequences, the so-called BRC repeats of BRCA2, and a single molecule, the core catalytic domain of RAD51. The point is that the interaction between the BRC repeat and RAD51 actually blocks the capability of RAD51 to oligomerize on DNA substrates. And if you'd been listening to what I said, you'll realize that oligomerization is a fundamental characteristic of RAD51, which enables it to form this ordered helical nucleoprotein filament. So what is a protein like BRCA2 that promotes RAD51 function doing by antagonizing, apparently, the ability of RAD51 to oligomerize on DNA substrates. It took us, believe it or not, 10 years to work that out. Mahmood Shivji in my lab expressed and purified this entire middle section of BRCA2 containing each of the RAD51 binding motifs, over 1,000 amino acids long, that fragment. And what he was able to show is that BRCA2 has opposing activities on RAD51 oligomerization on single-stranded DNA versus double-stranded DNA. In a nutshell, what we found was that in the absence of BRCA2, RAD51, when given single-strand and double-strand substrates, goes to the wrong one. It forms stable structures on double-stranded DNA, which is the wrong step. It's the second step, instead of forming stable structures on the single strand. What BRCA2 does is to stabilize the single-stranded structure, but inhibit the double-stranded structure. So if you will, it orders the reaction so it can proceed in the correct direction. We still don't understand the structural mechanism for this, but very recently, Jude Short, Mahmood, Xiaoxia Chen, and Yang Liu in my lab have been using cryo-electron microscopy to resolve now at about 3.5 to 4 angstroms the details of the RAD51 filament on single and double-stranded DNA. And suffice it for me to say, we still don't understand the structural mechanism, but we're beginning to get clues. So the point I really want to make is we now understand in some detail how loss of BRCA2 can cause genome instability and give rise to cancer by inactivating this critical pathway for double-strand DNA break repair. In fact, it's disruption of homologous recombination that also accounts for the therapeutic sensitivity of BRCA-deficient cancers to new targeted agents like PARP inhibitors or even to old drugs like platinum compounds. The repair of DNA breaks by HR is what makes normal cells resistant to the effect of these agents. So in BRCA2 deficient cells, this pathway being lost, cells are highly sensitive. So I've just realized that I've lost a number of slides, so I hope you'll bear with me. Yeah. Hundreds of variants of BRCA2 are inherited in the germline, which can give rise to cancer. And the point I want to make on this slide is simply that the majority, about 75% of these germline pathogenic alleles, work by truncating the BRCA2 protein. So how does BRCA2 truncation cause carcinogenesis? To address this issue, we decided to develop a series of genetically engineered mouse models to begin to look at tissue-specific carcinogenesis associated with these types of truncating BRCA2 mutations. We chose to do this in the pancreas, and the reason is quite simple. Unlike in the breast or in the ovary, we actually know the oncogenes that trigger oncogenesis in the pancreas. 90 to 95% of pancreatic ductal adenocarcinomas harbor an active mutant of the KRAS oncogene, so we were able to create a model in which KRAS was activated on one allele only in the pancreas, P53 was inactivated only on one allele, only in the pancreas, and we could either model the germline allele of BRCA2, a truncating allele which is found in all tissues, or specifically in the pancreas, we were able to engineer loss of heterozygosity when necessary, loss of that second allele. So if you think about it for a second, this mouse model is a little unusual in that typically people studying tumor suppressors just knock out both copies of the protein. What we have tried to do here is to recapitulate the events that occur in human carriers. A germline allele present in all tissues which truncates the protein, and loss of heterozygosity only in the target tissue, in this case, the pancreas. And what we found surprised us. What we found, and this is work going back about 8 years, done by Ferdinando Skoulidis, an MD PhD student in my lab at the time, was that loss of heterozygosity was unnecessary for carcinogenesis in this model system associated with BRCA2. Mark that. You don't need to lose the second allele. None of these mouse tumors had lost the second allele. They still got accelerated development of pancreatic ductal adenocarcinoma. It's not just a mouse artifact, because we were able to collaborate with Jon and Jorunn at the Icelandic Cancer Registry, where a large number of people in the population inherit this BRCA2 truncating founder mutation, and again we found that about 75% of pancreatic cancer arising in this population did not lose the second allele. And interestingly, but just as you'd predict, we were able to show that cancers in which this second allele persists are resistant to all the new targeted agents like the PARP inhibitors, as you'd expect, because the second copy is still functional. Recent work using genome-wide surveys of tens of thousands of cancers has confirmed our early results. This is again the WASH-U study, and what you can see here too in a study, as I said, of about 10,500 cancers, is that BRCA2 persistently violates the Knudsen two-hit paradigm. In the breast and in other tissues, between half to 60% of BRCA2 deficient cancers associated with these pathogenic germline alleles do not lose the second wild type copy. This is a study from Kate Nathanson and Susan Domchek, and what they find again is that about half of their breast cancers in families carrying the BRCA2 mutation do not lose the second allele. So how does this work? Well, the first question we asked was, could it be that heterozygosity has some subtle, not fully penetrant effect on DNA repair by homologous recombination? In the interest of time, let me say no. These cells have no detectable defect in homologous recombination, and as I said before, they are fully resistant to PARP1 inhibitors and platinum compounds, which trigger cell death in cells lacking homologous recombination. They also lack the genomic signature typical of DNA repair defects in homologous recombination. So how does it work? Well, what I'd like to share with you is the bottom line from some studies which we concluded just a few months ago, which show, in fact, that there is a mechanism wherein we believe the environment cooperates with the genetic inheritance of truncating alleles in BRCA2, and these factors conspire to induce BRCA2 haploinsufficiency. And what I'm going to share with you very briefly for lack of time is that there's a chemical trigger that we find that leads to the induction of haploinsufficiency in cells which carry one truncated copy of BRCA2. The chemical trigger is aldehydes, and as many of you know, these are found everywhere. They're products of endogenous metabolism. They're also found, I have no doubt, in much of what we've been inhaling in our trips to and from the India Habitat Center, because they come from car exhaust fumes as well. But for lack of time, what I'd like to share with you is what we found, rather than take you through all the steps in the science. What we were able to show is that aldehydes, whether synthesized endogenously or through exposure from the environment, induce a proteolytic action on a very small number of human proteins, about 50, one of which is BRCA2. This is a ubiquitin-independent but proteasome-dependent mechanism of proteolysis, and the net effect is to reduce the level of BRCA2 protein. Wild-type cells are also susceptible to this effect, but because they have more BRCA2 to start with, they're able to tolerate the effect of aldehydes quite well. BRCA2 heterozygous cells starting with half the amount of protein, although they're perfectly functional at that point, rapidly lose function. And this, if you will, induces haploinsufficiency. In other words, an environmental trigger induces haploinsufficiency in cells carrying this germline susceptibility allele, and that leads to chromosomal instability and, we think, carcinogenesis. Of course, there are many implications, not only for the potential for tissue-specific occurrence of aldehydes and tissue-specific BRCA2 deficient cancers, but also potentially opportunities for intervention through limiting or altering the way that these aldehydes are made. And I think many of you sitting in the audience will know that many metabolic processes ranging from diabetes onwards cause, if you will, changes in the way that many of these aldehydes are made in cells. So really what I'd like to convey is that diving deep into trying to understand heterozygosity and how it causes BRCA2 deficient cancers, we've found that BRCA2 can cause carcinogenesis without loss of heterozygosity, and discovered what we think is an interesting new mechanism that may underlie this. The thing to remember as well is that if so many BRCA2 deficient cancers don't lose the second allele, the PARP inhibitors are not going to be effective. And indeed, clinical experience suggests that about half of cancers are not sensitive to these types of inhibitors when they should be. And we wonder whether lack of LOH may be a contributory factor to primary resistance in this setting. So I've taken you, I'm afraid, in a rather whistle-stop fashion through some of our work on looking at germline alleles and how understanding the pathways for carcinogenesis may suggest strategies for early intervention in cancer. If I have 2 minutes, okay. I will just talk about one aspect of the work which is beginning to take place actually in India, which is why I wanted to share it with you. So many of us are beginning to understand and identify biochemical pathways that mediate disease. How does one begin to dissect these pathways to identify potential targets for intervention and to create the types of tools that can be used to test these ideas in the laboratory? So for the last 7 years or so in our lab in Cambridge, we have developed 3 groups of tools which integrate, we think, to help us to address this issue. The new methods to undertake phenotypic screening using genetic tools that essentially identify protein shapes which are involved in key phenotypic outputs. The ability to create, using structure-guided methods, chemical leads that target these types of shapes that mediate cellular functions. And finally, new kinds of microscopy that enable us to analyze the perturbation of cellular pathways after adding chemical tools of this kind. And this integrated suite of tools, we think, can help us to dissect disease mechanisms and potentially identify and validate actionable targets for intervention. This is some of the work that's actually occurring on structure-guided lead discovery at the Center for Chemical Biology and Therapeutics at INSTEM and NCBS in Bangalore. What I'd like to really end on is the answer, I hope, to what we had considered an important issue, which is cancers start from a simple scrambling of pathways to very complex traits. Can one intervene early by hitting early events to cause the loss or death of cancer cells? We think we can. And this is an example of using the types of methods that I just spoke about to develop selective chemical tools that target protein-protein interactions of a group of mitotic kinases which kills quite selectively cells which have nothing wrong with them other than that they express mutant forms of oncogenic KRAS. So for us, this has been not only a proof of principle that one can potentially intervene early in these types of settings to eliminate cells carrying oncogenic mutations, but also that the systems that we have developed to enable the discovery of these targets and the creation of chemical tools can work. So I'll stop there and say that the work that I've shared with you has largely been carried out at the MRC Cancer Unit in Cambridge, and the single slide I showed you about the work of the CCBT in Bangalore. Thank you for your attention, and I apologize for going over time.
We can have very brief 1 or 2 questions.
Yeah, I'm just getting some water.
Hi, Ashok here. Very nice talk. I'm very glad that I was able to hear this. Followed your work over the years, and one thing that is slowly at least becoming apparent to me is BRCA2 RAT51 interaction is probably a primary event in making sure that RAT51 engages the single strand. Am I right with that? So, and because breakpoints are going to be a sort of a, will not choose where the breakpoint happens, it appears that there's no sequence context. So the question is, do you think that RAT51, this interaction along with has the DNA sequence got anything to there? Or it is absolutely, doesn't care about what kind of DNA it's.
That's a good set of questions. There's more than one, so let me take them very briefly. The first is, yes, you're right. We find that in physiological ionic conditions, that without the BRCA repeats of BRCA2, RAD51 is unable to form stable complexes on single-stranded DNA. You can manipulate that a little biochemically by adding high concentrations of calcium or ammonium ions, but in physiological ionic conditions, you need BRCA2 to form that single-stranded filament. Second part of it is sequence specificity. You're right, there is no apparent sequence specificity for the repair event, but we don't know whether the break event that necessitates the repair is sequence specific. So many of the RAD51 associated lesions, BRCA2 associated lesions, occur during replication. And as you well know, there are many sites in the genome that are somewhat refractory to the transit of the replication machinery, so-called replication fragile sites, the late replicating regions, which tend to be in typically heterochromatic sites. And we wonder, but we don't know yet, whether these may be more susceptible to the breakage that then leads to mutation in the absence of BRCA2.
Hello. I have a question here. It's about aldehydes causing... Here.
I'm sorry, I'm trying to... Okay, got it.
Aldehydes causing haploid insufficiency due to a proteolytic event. So can inhaled aldehydes cause an increase in such proteolytic events, number one? And number two is, are you talking about the environmental aldehydes or like endogenous aldehydes produced during oxidative stress or something like that?
The second question is a straightforward answer, both. So what we're able to show is that exogenously added aldehydes, but also increased endogenous aldehydes, which, as you correctly pointed out, are generated during processes like, for example, lipid beta peroxidation, or even histone lysine demethylation. Many of the KDM demethylases produce formaldehyde as a byproduct. Many of these events can generate aldehydes that have an effect on BRCA2. Does that answer your first question as well? We don't know. It's certainly a possibility, which I alluded to in the context of the Delhi traffic, but we don't know for sure.
So
yes?
They say that half of our textbooks may be wrong. We just don't know which half, and I think you have shown a great illustration of a hypothesis we have held dear to heart for a long time. My question is that you have shown that the LOH is not necessary, but it still happens in half the cases. Clinically, do you see there's a difference in the patients that have LOH and others who don't?
That's an excellent question, and we don't know the answer. So basically, what we have been able to show is that the truncating mutations work by this induced haploinsufficiency pathway, we believe. What I didn't have time to share with you is that we've also found other mechanisms affecting different missense mutations in BRCA2. So we're in the process of working with collaborators across the world to identify families who carry those mutations to ask the questions which you're asking, which is, do they develop cancer earlier than the others? Is there LOH? Is there less LOH? Is there tissue selectivity? How do they progress clinically? We don't yet have the data to answer that. But let me make one point in relation to the haploinsufficiency induced by aldehydes. As I think many of you know, about 500 million people across this planet, 500 million, primarily of East Asian descent, inherit mutations in ALDH2, aldehyde dehydrogenase 2. This is the enzyme that catabolizes acetaldehyde after ethanol exposure. This is why people carrying this mutation flush, go red, they can't tolerate their drink well. It's intriguing to speculate, and we're trying to test this idea, that if they also carry BRCA2 mutations, that there may be, a working together, if you will, of these genetic alleles to somehow modify cancer progression, and this is something that we're testing.
I was just wondering that in terms of the early intervention, so if you have to do a trial, a clinical trial for an early intervention in cancer, like the holokinase what you showed, so what should be the endpoints? Because it's an extremely challenging trial to do.
You're absolutely right.
The endpoint measurements and everything is so complicated.
You're absolutely right, and I think that this is a major, major challenge for testing any potential drug-related early intervention. So the way we envision this is actually to intervene at a later stage in order to try to prove the concept. So for example, if we develop drugs which may help to eliminate RAS mutant cells, one can envision situations, for example, people who have pancreatic cancer, who have inherited mutations in the pathway, who are treated, to treat them as a secondary intervention opportunity. In other words, to see if you can delay relapse or recurrence or post-surgical grow out as a way to test in a shorter term the proof of concept for longer term intervention trials. I don't have an answer for the actual primary intervention trials. As you know, with tamoxifen, Rolodex, and the aromatase inhibitors, it's taken years and literally tens of thousands of patients to test those ideas. I think all we can do is to try to see if we can find situations in patients who have been treated and are at the risk of relapse or developing other forms of cancer, whether these types of treatments can delay or prevent progression. That's our idea anyway.
Thank you very much, Ashok.